This protocol enables scientists to quantitatively compare the number of transcription factor complexes under different shear stress conditions and will be beneficial for research in vascular biology. The main advantage of this protocol is the usage of microfluidic flow channels that allow investigations of several antibody pairs, including the respective controls, simultaneously. To begin, coat the 6-channel slide by adding 0.1% porcine skin gelatin prepared in PBS into the channels for 30 minutes at 37 degrees Celsius. Empty the channels from the gelatin solution and seed HUVEX in the pre-coated 6-channel slides at a density of 2.5 times 10 to the 6th cells per milliliter in 30 microliters of M199 complete medium. Allow the cells to adhere for 1 hour at 37 degrees Celsius in the humidified incubator. Add 60 microliters of pre-warmed M199 complete medium to each of the reservoirs. Culture the cells at 37 degrees Celsius for 2 days in a humidified incubator with medium replacement after 24 hours. Mount the silicon tubing on the fluidic units. Fill the reservoirs with a minimum of 10